Chapter 10. Cell Reproduction. All sexually reproducing living organisms start life as a fertilized egg known as a zygote. Afterwards, billions of cell divisions must occur to produce the young offspring from this zygote and then continue through juvenile stages and adolescence. Humans, like most mammals, have determinate growth, meaning they stop growing at adulthood. So while growth is one reason why cells divide, cells must also divide to repair damaged tissues and organs, as well as replace aging, dead, or lost cells. Cell division is highly regulated by enzyme activity and hormones. Failures of regulation during the cell cycle can lead to a fatal class of diseases called cancer. One of the basic principles of cell theory is that all life is made up of cells and that cells come from pre-existing cells. The cell cycle describes the life of a cell starting as a mother cell and ending as a daughter cell, which then becomes the mother cell of the next generation. Before we discuss the cell cycle, we need to go over some basics of genomic DNA. A genome is a cell's DNA packaged as a double-stranded molecule. However, it differs based on whether a cell is prokaryotic or eukaryotic. If prokaryotic, DNA is found as a single circular structure. This DNA is often called a chromosome and is housed in the nucleoid region, which is not a true nucleus, but nucleus-like region inside of the cell. Prokaryotes can also have independent circular chromosomes that have nothing to do with growth and reproduction. These are called plasmids. These carry instructions for other actions. The most studied are those that cause antibiotic resistance. In eukaryotes, there are several double-stranded linear molecules of DNA that we call chromosomes. Each species of eukaryote has a specific number, but it's possible for two species to have the same number of chromosomes. Humans have 46 chromosomes within their somatic or non-sex cells, but we also share this number with an amphipod and a species of antelope. So while chromosome number varies by species, it's the DNA within the chromosomes that indicate what species an organism is. Humans have 23 chromosomes in their gametes, or sex cells. When working with chromosomes, we use the letter N to represent the number of types of chromosomes present. N is an organism's haploid number. In humans, it's 23. However, most sexually reproducing organisms have 2 copies of each chromosome inherited from their parents, so we designate these 2N. In humans, 2N is equal to 46. In other words, 46 is a human's diploid number. During fertilization, a haploid sperm meets a haploid egg to create a diploid zygote. During this time, all homologous chromosomes, that is, chromosomes of the same size and containing the same genetic information, pair up. So mom's chromosome number 1 matches with dad's chromosome number 1. Mom's chromosome 2 matches with dad's chromosomes number 2, etc. Chromosomes house genes, which are made of specific nucleotide sequences that encode for a specific trait. There are variations of a gene called alleles, which means that offspring could display similar or different characteristics to one or both parents depending on the pairings of these alleles. Eukaryotic chromosomes are linear in shape. In humans, we have 46 total, which makes up 23 pairs. In essence, all of these chromosomes make up all of our DNA, which laid end to end would be 6 feet in length. But this molecule is so thin, it's only 2 nanometers in width. Because of this length, it must be tightly compacted within a microscopic nucleus of a cell. This compaction happens by having DNA spool around special histone proteins. When woven over and over, they create chromatin. But before cellular division, this chromatin becomes more condensed into chromosomes. After replication, the chromosomes decompress back into chromatin. The reason that chromosomes cannot be permanently in a tightly compact linear shape is that it needs to be readily accessible in case a cell needs to build proteins and lipids. Instructions for these are found within the DNA of cells. Chromosomes are copied into pairs called sister chromatids by the time mitosis begins. Each sister chromatid is an identical copy to each other. They are joined together by the centromere region by a kinetochore protein. This kinetochore protein contains attachment sites for spindle microtubules to attach and pull the sister chromatids apart. The cell cycle is a series of steps which explains how a mother cell eventually divides into two identical daughter cells. In this chapter, we'll be focusing on the asexual means of reproduction, mitosis, and binary fission. Cells are either in interphase or the mitotic phase. It's also important to point out that in adults, nervous and muscular tissue do not undergo reproduction once matured. Once nervous or muscular tissue is lost, it cannot be replaced. For other cell types, the cell cycle varies as a function of the cell's physiology. For example, skin cells replicate rapidly. Red blood cells last a few weeks before they need replaced, and some cells like memory immune system cells can last decades remembering old diseases. Interphase is when a mother cell carries out normal cell functions, but is also preparing for mitosis by duplicating DNA and cellular components. The vast majority of a cell's life is spent in interphase. Mitosis is when the mother cell begins to divide the DNA and components of these cells. And finally, cells undergo cytokinesis, which is the division of the cell membrane that forms two daughter cells. The reason why cells must divide into two rather than continue physical growth is because the larger the cell, the less surface to volume ratio it has, meaning that it will be less energetically efficient to carry out biological processes. Dividing into two cells helps to resolve this issue. So in the next few slides, I will demonstrate the cell cycle, and I want to introduce the important components involved. Many organelles are not shown here for the sake of simplicity. The plasma membrane, which is the border of the cell, is represented by this tannish-colored circle. The purple-colored circle is the nuclear membrane. The nucleolus is the dark purple circle. It's not really necessary to the process of cellular division, but it's still shown because it's part of the nucleus. You also have chromatin. Chromatin is going to be represented by these squiggly lines you see here inside of the nucleus. And finally, centrioles, represented by these orange rectangles. These produce spindle fibers, which are used to help to divide the chromosomes to each potential daughter cell and help split the cell. During interphase, the cell undergoes normal growth processes while preparing to divide into two daughter cells. Interphase is broken down into three stages: G1, S, and G2. Cells must successfully move through these stages in order to move on to mitosis. The G1 phase, or first gap phase, is called such because at the molecular level, the cell is preparing the DNA to be replicated and making excess ATP to prepare for replication. You might have also noticed that it physically begins to grow. The S phase, also known as the synthesis stage, is where the DNA is duplicated. Right now, the DNA is organized into chromatin, which is less condensed in chromosomes. When S phase is complete, there are two copies of DNA, which will eventually become sister chromatids in prophase. This is also where the centrosomes, which are important components of the cytoskeleton, start to replicate. To help prepare for division of a cell into sister chromatids, centrosomes give rise to the mitotic spindle, which will help the cell divide. The G2 stage occurs after the S stage. Here the cell must replenish any lost ATP needed to replicate DNA, so now that it is prepared for mitosis and cellular division. Also, you'll notice organelles become duplicated and the cell continues to grow. Before the cell can enter mitosis, it must pass some checkpoints related to proper DNA replication and ensuring that the cell has the proper resources to continue on to mitosis. If everything's good to go, then the cell enters the mitotic phase. If not, the cell will enter a period of stasis or be destroyed, but we'll look at that later. The mitotic phase is split into two parts and 5 phases. Karyokinesis takes place in 3 phases: prophase, prometaphase, and metaphase. The purpose of karyokinesis is to divide DNA in the original nucleus. Cytokinesis takes place in 2 phases, anaphase and telophase. The purpose of cytokinesis is to split the cells into 2. The first stage of mitosis is prophase. During prophase, chromosomes condense and first become visible under a microscope. The nuclear envelope disintegrates into small vesicles, and membranous organelles like the Golgi complex and endoplasmic reticulum will start fragmenting towards the edge of the cell. The centrosomes begin to move towards the opposite ends of the cell. Then it moves on to prometaphase, where more disintegrating of the nucleus occurs and the mitotic spindle further develops. Chromosomes also become more condensed with sister chromatids, the centromere or area where the chromatids attach. The mitotic spindle is made up of microtubules called spindle fibers, and there's two types. Polar microtubules, their job is to push the poles of the cell away from each other during mitosis, and kinetochore microtubules will pull the chromosome, the sister chromatids, away from each other and towards the poles during mitosis. During prometaphase and metaphase, the formation of the mitotic spindle is complete. Motor proteins on the kinetochore microtubules pull each chromosome in opposite directions, causing the chromosomes to line up in the middle of the cell. The imaginary plane formed by this is called the metaphase plate. At the end of metaphase, karyokinesis ends and cytokinesis begins, which then leads us to anaphase. And during anaphase, centromeres split and sister chromatids are pulled by the spindle fibers toward opposite poles of the cell. Replicated chromosomes split into two identical sets of unreplicated chromosomes, and as soon as they are no longer attached at the centromere, sister chromatids become daughter chromosomes. In addition, motor proteins of the polar microtubules push the two poles of the cell away from each other. So next is telophase. During telophase, a new nuclear envelope begins to form around each set of chromosomes, and the mitotic spindle begins to disintegrate. Chromosomes then begin to decompress back into chromatin. When two independent nuclei have formed, mitosis is complete. Now we must complete the division of the cell. So cytokinesis typically occurs immediately after mitosis. During this process, the cytoplasm divides to form two daughter cells, each with its own nucleus and complete set of organelles. Remember, cytokinesis actually begins in anaphase and it completes after telophase occurs. Cytokinesis in animals, fungi, and slime molds occur when a ring of actin and myosin filaments contract inside the cell membrane, and what it does is it causes itself to pinch inward, forming a cleavage furrow. This causes the cells to divide into two. Cytokinesis in plants is going to be slightly different, whereas the other stages of the cell cycle are pretty much the same as animals. In plants, because they have a cell wall, the way it divides will be a little bit different. So cytokinesis in plants occurs as vesicles are transported from the Golgi apparatus to the middle of the dividing cell. These vesicles fuse to form a cell plate. Not all newly formed daughter cells immediately begin a new cell cycle. It's possible that cells can enter what's called the G0 stage, which means they're not actively dividing. As mentioned earlier, mature muscle and nervous cells in mammals eventually reach the G0 stage and never divide again. Other times, cells are affected by particular environmental conditions, such as lack of nutrients and other growth factors. This can place them temporarily in the G0 stage until conditions improve. So up next, we're going to talk about the control of the cell cycle, cancer, and how prokaryotes undergo cellular reproduction.